Hello students, hope you are doing well. So today we are going to discuss about antibiotic sensitivity. So first of all, we will discuss what are antibiotics. Antibiotics, as we all know, that these are chemicals that are produced by the bacteria or fungi that have the ability to prevent other organisms from growing or killing them. So these have been widely used in controlling bacterial infections in humans that cause diseases, and there have been many antibiotics that have been developed time to time. So, while there are many different kinds of antibiotic and still many more kinds of bacteria, not all antibiotics are effective in inhibiting all bacteria. So it is essential to determine whether an organism is sensitive to the inhibitory action of the antibiotic or not. So there are many procedures to determine it. So the most common and the modern technique used is disc diffusion method. So the procedure for this method is that filter paper discs are saturated with specific amount of antibiotic and placed on a growth of particular bacterial culture to be tested. So from this experiment, two conclusions can be seen. The first, growth is inhibited, then sensitivity is seen. Second case, if growth is seen, then the culture is resistant to the antibiotic action and is not inhibited, which will mean that if cannot be used to treat an infection by the bacterium. So, it has been discovered that antibiotics inhibit bacteria by stopping their growth or killing them. So, antibiotics have their different modes of action. So, the mode of actions can be destruction of the cell membrane, inhibition of protein synthesis, inhibition of DNA synthesis, and the last is inhibition of intermediary metabolism. So the most antibiotics are effective only against bacteria, and the disc diffusion method, like we have discussed, so we can take the two examples. First, we are taking E. coli that represent the gram negative organism, and hence the second example is about Staphylococcus aureus representing the gram positive bacteria. So from this we can conclude that antibiotics that are effective against a wide range of bacteria can be considered as broad spectrum, whereas those that have effect against a small specific group of bacteria are called as narrow spectrum. I hope you have understand this topic. Thank you so much.